Okay, perfect. So welcome to our BrainMap seminar today. It is my honor to introduce Dr. Hai-Ling Cheng. So Hai-Ling is an electrical and computer engineer by training. She worked in industry for Canadians defense surveillance before returning to academia at the University of Toronto to complete her PhD in medical biophysics. She is currently an associate professor at the University of Toronto in the Institute of Biomedical Engineering and Edwards Rogers Department of Electrical and Computer Engineering. She focused her research on novel molecular imaging technologies and MR contrast agents for cardiovascular diagnostics, therapy guidance, and regenerative medicine. She spent a decade at the Hospital for Sick Children, where she developed robust quantitative MRI platforms for clinical workflow. She has over 75 peer-reviewed publications and holds patents on novel contrast agents and cell tracking platforms. Hai-Ling and I met at the annual meeting program committee for ISMRM in 2020. In January, that was basically my last meeting where I saw people in person. And at the time, she was the table chair for the contrast mechanism table, and a role that I coincidentally took over, realizing how much work that really is, especially so right now we are in crunch time as well for ISMRM. So today, Dr. Hai-Ling Cheng will talk about new MRI platforms for cell tracking and tissue engineering applications. So please take it away.
Thank you for the kind introductions, Eva. So as Eva had already mentioned, I worked at the Hospital for Sick Children before joining the University of Toronto, the Faculty of Engineering. And the work that I'm going to talk about today really started in earnest in around 2015, 2016. I was surrounded by colleagues who were interested in regenerative medicine using stem cells, and colleagues who were making biomaterials to build 3D tissue structures. And so I realized very quickly that there was a clear need for imaging, specifically MR, to help advance these fields. And this is what I'm going to be focusing my talk on today. Slide. So when we think about implants or anything that we put into the body, it's constructive if we think about how we used to think of implants. And 20 years ago, if someone were to say to you, can you image an implant on MR? Most likely you'll think of a metallic implant, as shown in this example here, where there's a metallic metal sphere implant that is sitting at the top of the femoral joint. Depending on the type of MR acquisition that you apply, the susceptibility, blooming artifacts, that dark contrast can have various sizes and shapes, but undoubtedly it's very easy to locate where the implant is sitting, although not so easy to define its actual borders accurately. Now, when we think about metallic implants, of course, it's easy to image these because the material properties are so different from those of endogenous materials like cells and membranes. And metal isn't the only material that we can image easily on MR. Take, for example, this 54-year-old woman with a silicone implant in the breast. The implant had ruptured, and depending on the type of sequence that we use, shown here up in the upper left, is a silicone suppressing sequence. In B is a silicone enhancing sequence, and the extent of the silicone is indicated by these white asterisks. If we use a T2 or T1 weighted sequence, as shown in the bottom, you can easily identify not only where the silicon is located, but also where the silicon has leaked outside of the bag into the surrounding parenchyma. So imaging implants with very different material properties than endogenous tissue is relatively easy. But when we think about implantation these days, often we're not talking about metal screws or silicone. We're most likely talking about actual cells or materials, biomaterials that are designed to mimic the natural extracellular matrix. And a case in point is all of the regenerative medicine research that's going on around the world, as exemplified here by work from Prof. De Veer at Tel Aviv University, where very recently, in the past 2 years, he's demonstrated that he was able to take fat tissue biopsied from a patient's stomach, and he could take that fatty tissue, isolate the fat cells away from the extracellular material. For the fat cells, he would then reprogram them into induced pluripotent stem cells. And from here, you can go on to derive multiple different cell types: cardiac cells, neurons, spinal cord cells, etc. The material, the extracellular material that is separated during this process, could then be converted into a gel-like form, like a hydrogel. It doesn't have any cells in them, and it's basically there to support cell growth once it's put it back into the body and allow the cells to form functioning tissue. So with this type of workflow that's exemplified here in Prof. DeVere's lab, we can now generate cells of the desired type that we want and the extracellular matrix material for minimal risk of rejection after it's put back into the body. And with this workflow, we can put the cells and matrix material back into the body as is, or we can take them through a 3D bioprinting step to create not just a blob that's injected, but to create an organized 3-dimensional structure, such as a heart, as shown here. In fact, this lab at Tel Aviv printed the first ever 3D heart using this method, and this was reported back in 2019. Okay. So this is what we're talking about these days when we think about implantation. We're talking about cell implantation, we're talking about natural material implantation. And for today's talk, I'm going to break it up into 2 separate segments. First half, I'm going to talk about how we can track cells in vivo on MR. And then in the second half, I'll talk about biomaterial tracking on MR. So just a little bit of background on why we want to put cells into the body in the first place. Now, the most obvious reason, as I've already alluded to, you can take stem cells, whether they are embryonic or induced pluripotent stem cells. You can take these, grow, differentiate them into the cell type that you desire. Maybe it could be spinal cord cells, or maybe they could be cardiac muscle cells. And the purpose here is to introduce the cells into the native organ to grow new tissue. Okay? But cell therapy has other uses as well. For example, they could help to restore function, and not necessarily by growing more cells. For example, in diabetic patients, you might want to implant islet cells to secrete insulin to restore function. And in cancer therapy, you can inject immune cells to help kill off the cancerous tumor. Now, regardless of the purpose of the cell therapy, there are a number of obstacles that are shared amongst these different applications. And I'm going to list the most important ones here. Okay? First of all, mass cell death occurs every time after delivery. Sometimes the cells die because they are going through the needle. And a lot of times it's because the cells are introduced to an in vivo environment that's not conducive to their survival. You may be injecting cardiac muscle cells into an ischemic area of the heart. And therefore, in a lot of applications, between 30 to 50% of cells actually die within the first day. And after 1 week, easily over 90% of the injected cells could have already perished. Okay. And once you inject cells into the body, unless you had a non-invasive way of tracking them, there's really no means by which we can determine how many cells have died. And therefore, as I've listed right here, it's very difficult to ascertain cell retention. Now, the other thing I want to mention is that cells can also migrate. They could go to different areas of the body and exert mechanisms of action in places not only confined to the injection site. But if we don't know if the cells are surviving or not, and if we don't know where the cells have gone, it's really difficult to correlate improved tissue function, say, with the mechanism of action from the cells. So these are the primary difficulties to progress currently. And luckily, there are ways that we can, there are methods by which we can, that we can take advantage of to look at cells non-invasively. What I'm showing here is a table that I've taken from Eric Aaron's paper. It's a rather old paper, but still very nice table. And really the only technologies, the only platforms that we're interested in are the ones that provide unlimited penetration depth. Because if we want to be imaging human patients in, deep in the body, these are the modalities that we want to be focused on. And when we think about modalities that don't require an anatomical co-registration obtained from a different modality, that really means we have to rule out nuclear medicine, SPECT and PET, and really what we're left with is MR and CT. But if we think about the sensitivity of detection that we want to achieve, MR is really a very, very good candidate. And in the next slides, I'm going to give you a little bit of background on how this is done on MR traditionally and what we're doing now differently to make it even better. So with conventional cell tracking on MR, normally what we do is we take a contrast agent. It could be a T1 reducing agent, a T2 reducing agent, or a fluorinated agent, depending on the type of coils that you're using. And we use these chemical agents to label our cells. So we put the chemical agent into a cell culture dish, into the cell culture media, and we let the cells absorb the agent, whether passively, or we could do it by active means, such as electroporation. Once the cells have taken up the contrast agent, the cells are then washed so that any remaining agents in the media are eliminated, and the cells that are now labeled are injected into your subject, who then goes onto the MR scanner to be imaged using the appropriate atomic species, okay? Now, as far back as 20 or more years ago, people have already been doing cell tracking on MR, and they've demonstrated feasibility. In fact, when we look at this paper from 2003 from Dara Kratchman, they were able to take mesenchymal stem cells and label them with an iron oxide agent, Ferridex. These labeled mesenchymal stem cells were then injected into the left ventricular myocardium of pigs, and you can easily see this hypointense area indicates the location of cell injection. And so this was shown 20 years ago, and the question comes to mind is, why are we still talking about cell tracking today in 2022? And the reason is really simple, is because even though it's feasible, there are a couple of really severe, significant limitations with how we do things conventionally. The first problem deals with signal dilution. If you use a chemical agent to exogenously label cells outside of the body, the content of the contrast agent on a per cell basis will reduce every time the cells divide. So after a few cell divisions, you're not going to have enough contrast agent remaining in the cells on a per cell basis to give you the detection sensitivity that you need. Furthermore, a lot of contrast agents, as easily as they go into the cells, they could as easily come back out. So even independent of signal cell division, you can lose signal that way. But perhaps even more severe limitation is that of non-specificity. The cells that you see in here are iron oxide label mesenchymal stem cells again, that have been injected into the myocardium. So you can easily recognize the location of the injected cells by virtue of this hypointense region. And it's monitored out to 4 weeks post-injection. Now, when you look at these images, you would assume, and I would assume, that there's this hypointense region. So obviously the original mesenchymal stem cells must still exist, right? They must still be alive. But that was not the case. As these authors reported back in 2015, at 4 weeks after cell delivery, there were actually no living mesenchymal stem cells left. So they had basically all died out. But then the question becomes, why is there still a hypointense area? And what histology revealed was that on H&E, we could still see these blue spots, which represents areas of iron accumulation. And when you superimpose this slide onto the CD68 stained section, which stains for macrophages, you could see that the blue dots are co-localized with the brown dots. And what this means is that the original mesenchymal stem cells died, releasing their iron oxide, and it was the macrophages that came in that took up the iron oxide and are still giving this persistent hypointensity. So that is the non-specificity that we're talking about. Now, to overcome these challenges with conventional cell tracking, I'm going to talk about how we can do things over the long term. How can we track cells and look at them regardless of how many times they divide, and as long as the cells survive? Plus, we want to be able to know that the signal is coming only from the cells that we labeled and not from anything else. So the solution to this, one solution to this, is genetic reporters. Genetic reporters are not unique to MR. In fact, they are frequently used in optical imaging. And the idea here is that we take a particular gene that when we upregulate its expression, we actually can induce intracellular contrast generation. And when we get cells to give off contrast from inside the cell, that actually achieves several of our objectives. First, the contrast will only come from cells that are alive, not dead cells, because once the cells are dead, you really can't be creating any proteins. None of that can happen. And because the genetic reporter is encoded into the DNA, it doesn't matter how many times these cells divide, and the signal on a per cell basis should remain stable regardless of the number of cell replications. So this gives us the potential for long-term cell tracking of the viable cell population only. And in MR, the genetic reporter that's been most frequently cited is the ferritin system. Ferritin is a very ubiquitous iron storage protein. It's found in almost all living cells, including bacteria and prokaryotes, and even some higher order plants. And what it does is it stores away iron. Essentially, that's what it does. And because iron is a susceptibility producing MR contrast agent, that gives us a dark contrast on MR. All right. The question then is, why are we not seeing more of this ferritin MR cell tracking system being cited in the literature? And the reason comes down to insufficient sensitivity. So this paper, very well done by, I believe it was Chuck Murray's group, led by Dr. Naumova, where they compared the ferritin system, so shown on the bottom row, against exogenously labeled cells. Okay, so in this paper, what they did was they took mouse skeletal myoblasts, which is a precursor, muscle precursor, and they labeled it with iron oxide and then implanted it back into the myocardium, or they had these skeletal myoblasts and they simply overexpressed ferritin in these cells. So when you could easily appreciate that at the top, there's this really obvious region of hypointensity. Depending on the type of pulse sequence that we use, you could see it on every type of pulse sequence, large area of dark signal. Whereas for the ferritin system, you'll be really hard pressed to find that negative contrast region. And on histology, what they found was that when you look at Prussian blue staining for iron, in the exogenously labeled samples, you could see a lot of blue dots here, which is indicative of the high degree of iron accumulation, whereas there was a substantially lower degree of iron accumulation in the ferritin overexpressing system. So this lack of adequate sensitivity has been what's held reporter gene systems from really taking hold in the MRI world for cell tracking. And what we had come across just very recently in the past few years, this is work that we published right in the middle of COVID, and we managed to file a PCT patent on this last year in 2021, was using the ferritin system, we can actually garner a bright signal from it to produce a very sensitive and specific MR reporter gene. So what I'm showing you here is a representative mouse that's been injected in this right leg, left looking at the screen, with with these human embryonic kidney cells that overexpress the ferritin protein. The left leg, okay, or right in our case, has been injected with wild type human embryonic kidney cells. So no overexpression of ferritin, they're just normal cells. You could see that after the administration of a manganese contrast agent, we get significant bright contrast enhancement in the ferritin leg, but no increase in the contralateral control leg. And this bright signal persists until it's washed away after 7 days after contrast administration. The interesting thing that we've also noted about this is that when we compare our newly discovered Bright-ferritin system against the divalent metal transporter gene, which has also been reported for MR cell tracking. The DMT is shown here in the middle line. Our Bright-ferritin is here at the top. You could see that the change in the relaxation rate, T1 relaxation rate, is much, much higher for our system. And when we look at changes in the T2 relaxation rate, the T2 star relaxation rate, similar to the study from Nomova, we actually did not see any substantial difference visually nor quantitatively of the ferritin system compared to wild type cells. So this in vivo study really confirmed that the ferritin system doesn't have the sensitivity when you're working it alongside iron, but when you combine it with a manganese contrast agent, it gives you substantial bright contrast. My former graduate student, Daniel, then validated that nanoparticles had actually formed within these cells. And so what he has here are photographs of the cell, intact cell, where I've blown up this red region showing these dark spots, which are supposed to be these nanoparticles, okay, the ferritin particles, engulfed in some kind of vacuole, and we believe this to be a lysosome. So the ferritin manganese nanoparticles that are formed don't stay in the cell forever. They are recycled through the cell's normal recycling system. And when Daniel purified these cells, these ferritin overexpressing cells, and precipitated out what was left inside and did transmission emission microscopy, he found that there were these electron dense manganese ferritin particles that correspond to roughly the size of ferritin particles that you would expect to find in mice. Looking at the quantification of manganese, we found that with the ferritin overexpressing system, when exposed to a manganese contrast agent, we get over twice the uptake of manganese compared to wild type cells. And this roughly corresponds to a doubling of the ferritin overexpression in our system. So ongoing work in the lab by a new graduate student, Ku Shuang, has shown that this system where we're manipulating the ferritin expression does not seem to have an effect on the stemness of our human embryonic stem cells. So what we have here is a stain for two different markers, OCT4 and SC4. So OCT4 is an octamer binding transcription factor. It's a marker for undifferentiated cells, and it's very critical in the self-renewal of undifferentiated embryonic stem cells. So this simply says that we have stem cells that are capable of perpetual self-renewal. Okay? And you can see that there's no difference between the wild type and the mutant cell lines. The second stain, SC4, is a cell surface antigen on both embryonic and pluripotent stem cells. And again, it's a marker for stemness. The next test we wanted to do was to see if these cells, despite having their stemness property, could be differentiated into functional mature cell types. So we chose to differentiate our stem cells into cardiac muscle cells. Simple reason being that this is the first differentiated cell type that we are currently working on. And again, we stained for two different markers, the first being cardiac troponin T. This is a protein that's very important to the contraction of cardiac muscle cells. And you can see that there's visually really no difference between the two cell lines. And we also stained for sarcomeric alpha-actin, which is a protein that sits within the sarcomere, the smallest functional unit in cardiac muscle. So that concludes the first part. In terms of cell tracking, I'm going to now talk about the contrast agents that we also have been developing for tracking biomaterials. Now, when we think of biomaterials, it doesn't necessarily have to be a hydrogel, right? A blob shapeless form that we inject into the body just to help cells maintain their viability. Biomaterials can take many forms. You could be bioprinting a 3D biomaterial structure, or in its most complex manifestation, you can actually have an organ structure. And what I'm showing you here are decellularized whole lung heart segment from a mouse. Here is a trachea from pigs, bladder, and if we want, we could take these acellular matrices from animals, and we can churn them up into a hydrogel, an extracellular matrix hydrogel, if you wish to work with it that way. Okay, and the point here is that regardless of the form in which the biomaterial takes on, there are a number of different applications. You can use biomaterials without cells to help restore cartilage. You could use it potentially to treat cardiovascular disease or to replace breast tissue. So these are just a few examples of what can be done. But similar to cell tracking, there are a number of obstacles to progress. First of all, because these materials, especially the ones that I'm showing you here, are very similar to native tissue, if you put them into the body, it's going to be very difficult to identify where they are and what structure they have. And when we're using biomaterials, we're basically using it for one express purpose. We want the biomaterial to provide some form, some structure, while the cellularization progresses in vivo. But we also want the biomaterial to slowly degrade to make room for regeneration. If it's degrading too fast, then the cellularization process might not be able to keep up with it. And if it's degrading too slowly, it can also interfere with regeneration. So being able to determine how fast these materials are degrading inside the body is really important. And it's not easy to estimate how they degrade from in vitro studies. Okay. Also, we want to look at how the scaffold remodels as a consequence of putting cells on them or an exposure to drugs. So let's look at some conventional methods that people have reported for biomaterial tracking on MR. Similar to cells, iron oxides are very commonly used because they are attractive. They provide a lot of sensitivity. So in this diagram here, we have a bunch of hydrogel that's been labeled with iron oxide. And in the top two images, what we have is hyaluronidase to help degrade the hydrogel. So when you use a higher concentration of this enzyme, the degradation happens more quickly. When you use a lower concentration, the degradation happens more slowly. And if you don't have anything, no enzyme, it doesn't degrade, okay? At least not noticeably. And you can see that because the hydrogel is labeled with an iron oxide particle, as it degrades, the dark area, the extent of dark signal starts to shrink. And you can use this to tell yourself how fast it's degrading. But it doesn't give you a good delineation of the borders of your biomaterial. And so more recently, people have turned to not iron oxides, but T1 agents like gadolinium is shown here for labeling their hydrogel such that they don't have this blooming artifacts that obscures all the anatomical regions around the hydrogel. So this is not an in vivo study. What they did was they took a hydrogel that was labeled with gadolinium and they injected it into a chicken wing. And you can see that the GAD labeled hydrogel has much brighter contrast compared to non-labeled hydrogel. I guess the main concern here is that when you're working with gadolinium, unless you can have a way of ensuring that it's very safely and strongly chelated, there's always the potential that the gadolinium can demethylate and come off of its chelate. And this is a particular problem when we're working with biomaterials, because biomaterials are meant to stay in the body for days and weeks and maybe even months. So the potential that gadolinium can come off over that time frame becomes much greater. It was for this reason that my lab started to look at safer T1 contrast agents for the purpose of monitoring biomaterials over very long time courses. So what I'm going to present to you over the next few slides are our results that we have actually published on in the past 3-4 years, very recent. This first slide shows how we are able to label hydrogels made of collagen with a manganese-porphyrin MR contrast agent. So the manganese-porphyrin contrast agent offers one very big advantage in terms of stability. The porphyrin ring is very thermodynamically stable, so there's very little chance that the manganese ion will come off. And even if it did, don't think it will, but even if it did, the body naturally has small amounts of manganese. So unlike gadolinium, our body has manganese, and very small amounts are not toxic to the body. But that's not a concern because we know that the porphyrin ring has very high dynamic thermal stability. Okay. Now, what we've done is we've taken our manganese porphyrin contrast agent, and we came up with a way for labeling collagen hydrogels such that it stays for days and days and days. So along the top row is an example of a labeled hydrogel injected in the subcutaneous space on the back of a rat. And over the course of 3 weeks, you could see that the bright area, the extent of the bright area has shrunk significantly. The size of this, as measured on MR, corresponds to the size of the scaffold upon explantation. So this tells us that, we expected this. This is not a surprise. But I guess what is more telling is that when you compare it against the unlabeled collagen hydrogel scaffold that's also injected into the back, you get this strange dark signal on day 1 only. And we believe it's because of the microstructure of the collagen. But soon after, that dark signal disappears and you really don't have anything to go by. It's impossible to tell if the scaffold has degraded. But 2 weeks after implantation, because we saw nothing, we wanted to confirm that the scaffold was still there, and there it is, still remaining, but you can't see it on MR. Now, what I didn't show you were some very gruesome images where we took apart, we sacrificed the animal and tried to explant the scaffolds, and there was nothing there. And it wasn't until we went back to look at the MR images that we discovered some animals, for some scaffolds, the scaffolds actually migrated from the back of the body to the front of the neck. And if you look at the MR, you could clearly see the migration. But if you open things up surgically, I would have come to the conclusion that the scaffold had degraded when in fact it had not. So it just highlighted the importance of having this ability to visualize where your scaffold is and if any unintentional migration had taken place. Now that was for a hydrogel. The next objective we tried to achieve, and we successfully achieved this, was to see if we could take organs from animals, strip away the cells to make them acellular or decellularized, and still be able to use the contrast agent to label them. So here, as I've shown before, are the decellularized intact organs from various types of animals. And after labeling, they take on this distinctive spinach-like color, and this is how we can visually know we've got it stained. The scientific question we were trying to pursue here was, even in the presence of a dense extracellular matrix that is left behind after this decellularization process, are we still able to label the construct appropriately and sufficiently? And so I'm going to show you a few slides on what the MR images look like. This is a pig trachea completely stripped of cells, and we took our contrast agent to label it in different ways. Sometimes we would label just the interior of the trachea. Sometimes we label it from both inside and outside. And for some of them, we label just the central part of the tube. As you can see on these T1 weighted images, the region of enhancement accurately tells us where that label exists. And this result is significant for a few reasons. First of all, trachea is made up of really, really dense cartilage. And the fact that the region that we selected for labeling could be selectively labeled and could be clearly demarcated on MR means that this workflow and this contrast agent works for labeling decellularized organs. Now, the next organ that we tested was the decellularized heart-lung block from a mouse. And you can see it's all greenish after labeling. On MR, we don't have any label on it. It just looks like nothing is there, right? It's against native background tissue. If we label it, it's all good, right? Really, really bright signal. What is interesting here is that not only can we see the labeled heart and the labeled lung tissue really well and see all the fine structures within it. Don't forget, this is a mouse imaged on a 3 Tesla human-sized scanner. Okay, it's not a preclinical scanner. But the important takeaways here is that our contrast agent was able to penetrate and bind throughout the heart tissue very efficiently. And if you look at the density of the bright contrast, that density, that brightness corresponds to how dense the signal, the tissue actually is. So if you're looking at the myocardium of the heart, of course, you've got a brighter signal because that tissue is much denser when compared to the lung tissue. And of course, whenever you're working with contrast agent, the most important thing to assess very early on is if it's cytotoxic, does it kill your cells? What we've shown here when we labeled human umbilical vein embryonic cells is that the labeling concentration of our contrast agent doesn't affect either the metabolism or the proliferation. In fact, we noted that there was a significant increase on our metabolic assay when we're labeling it compared with no label. And after some really careful observations, we discovered it was because our contrast agent not only did not kill off cells, they helped the cells bind better to the cell culture plate, which was the reason why these readings were all higher in the presence of our contrast agent. And then lastly, on this study, we wanted to see how long the contrast agent would stay in the scaffold. Full well knowing that we did not purposely design for covalent binding. Any binding that occurred between the contrast agent and the tissue and the scaffold is mostly through electrostatic forces. And so here on the left are some transmission emission tomography images of our decellularized scaffold. So here we've got our decell scaffold. Here we don't have anything, it's simply a blank grid. And you could see that our contrast agent, okay, represented by these black dots here, indicated by these red arrows, they do form clumps, but only in the presence of the decellularized matrix material. There's the particle, the contrast agent doesn't adhere to anything in the presence of just a blank grid. And the other point is, how long does it stay? Well, when we looked at the mouse lung, decellularized mouse lung, and we quantified the amount of contrast agent that had leached out over the course of 1 month, 30 days, we found that over 80% of the initial contrast agent stayed up to day 30. And any contrast agent that was released, it happened over the first 3 days after labeling. So this tells us that the stability is there, despite the fact that these agents were not designed to covalently bind. Now, this is work that's going to be coming out very soon. But what we had done was, given the successes that we had with the contrast agent for biomaterial labeling, we wanted to see if we could put it through a bioprinter. So a more recent grad PhD candidate in my lab, what he'd done was he took a gelatin alginate mixture to create a scaffold. It's a very simple geometry. We just wanted to prove it for proof of principle. So after bioprinting this really simple mesh-like layer structure, he labeled some of these scaffolds with the contrast agent and some without. And you can see that on MR, on a T1 weighted scan, the labeled printed scaffold up here at the top is very, very bright, has a correspondingly very low T1. I recall it's about 200 to 300 milliseconds in T1, versus very, very high T1 of obviously a gelatin alginate mixture. So clearly he was able to come up with a method, a bioprinting method that could take this contrast agent and still incorporate it into the bioprinting workflow. And really to test this in vivo, he took the labeled mesh and also unlabeled meshes and implanted them subcutaneously onto the back of a rat. And again, you could see the clear delineation on MR. The clear delineation on MR that's coming from the labeled scaffold. And on the unlabeled scaffold side, it's really impossible to tell where the borders are, if it had shifted. There's really no information because the material is just so similar to native tissue. Now, all of this work that I've just presented on biomaterial labeling was designed with no covalent binding in mind. And even though we had evidence that our contrast agent would last for a month, it's still much better if we could come up with a way of actively targeting our biomaterial. So over the past 2 years, my lab has been working on a family of new contrast agents, still based on the manganese porphyrin, but agents that are able to covalently bind to a variety of biomaterials, whether they are natural or synthetic polymers. And just to show you what can be achieved with this, here what we have, this bright area here is a polyurethane polymer biomaterial scaffold that one of my grad students created. He used one of our newer contrast agents. This is all confidential data. We haven't published yet. And compared to the control scaffold with the unlabeled, no labeling, you could see, again, a very distinct difference between these two. But I guess what is more interesting is that it's not until you actually have the label on the scaffold that you can see the structure and the boundaries of what the material looks like in vivo. And we never expected there to be this hollow gap in the center, but it existed. Now, without showing you what all the results look like, the most important result I'm showing you here is that when we're labeling a scaffold, of course we want the signal, but we also want to make sure that the labeling itself doesn't interfere with the degradation kinetics of the material. So we had a number of animals that were sacrificed, on a biweekly basis, and over the course of 7 weeks, what we had found was that the implants taken out from the control implants that were taken out from animals, represented by this blue line here, they degraded at the same rate as the labeled scaffolds that were taken out from the animals, as shown, reverse. Control is the orange, labeled is the blue. So you could see that when we look at the implant volume on histology, there's really no effect on the degradation rate, which is exactly what we had wanted. And most importantly, because this contrast agent that was in the polyurethane was sitting in the animal for 7 weeks, we wanted to be sure that there was no toxicity effect systemically. So we did a study of all the key organs, and we found that at 7 weeks post-implantation, the morphology of the liver, kidney, spleen, and lungs were all normal. There was no sign of inflammation, or there was nothing going on. Okay. So I'm just looking at the time. I want to leave time for Q&A. So in summary, I've presented MR reporter genes first as a good method for offering long-term and specific cell tracking of only viable cells in vivo. Unlike previous reports on MR reporters that suffer from low sensitivity, our Bright-ferritin platform gives very high sensitivity of detection for on-demand and very specific cell tracking. And by on-demand, what I mean is that when we want to recall that signal in the cells, from the cells in vivo at any time point after cell implantation, we would be able to do so via the administration of a contrast agent. Some future areas for exploration. We have yet to really carefully investigate the contrast to noise that's attainable in different organs and different cell types. We also need to look at the impact on the function of different cell types, not just cardiac muscle cells. And then depending on the cell type and the target organ, we need to really optimize the dosing of manganese, of the manganese labeling. And in regard to the biomaterial tracking work, I've shown you some results of our family of contrast agents that give us very high sensitivity, material tracking, and potentially with our new family of agents that target materials covalently, we are aiming to look at quantitative assessment of degradation. Now, the other thing I want to reiterate is that our contrast agents are able to label a wide variety of materials. This is actually in the process of being converted into a provisional patent at this time. So I hope to have more to report in the future. But most importantly, the contrast agents that I've presented can be adapted to a number of different workflows, and not only just for regeneration, but possibly for even drug delivery vehicles where you've got a drug encapsulated within a nanoparticle. You could perhaps label that nanoparticle, watch where it goes, and see if it's actually delivering drugs at the intended location. So with that, I want to thank everyone for your attention. I want to thank Eva for the invitation, and want to just welcome any questions at this time. Thank you.
Great. Thank you so much, Dr. Cheng, for this great talk. I'll just remind the audience to please use either the chat, Q&A box, or just raise your hand. Eva, if you have any questions, you're welcome to start.
The obvious question is, so what would it take to move this to humans? What are they? What would you guys need to do to prove that it's safe and all that stuff?
So so we haven't started anything in terms of the translation for the biomaterial tracking work. We've already started the legwork for the cell tracking platform, though. For either the cell tracking or the material tracking, these contrast agents are treated like a drug, and so there would have to be extensive toxicity studies. We'd have to get Health Canada approval and FDA approval in order to have this be applied clinically, and that's a long road. So what I started to do last year was I got a consultant. I got a consultant to do a market analysis on our cell tracking platform, and he interviewed. There were 25 interviewees from different academic centers, industrial centers, pharmaceutical companies, CEOs. And the review came back really positive. A lot of the reviewers had suggestions for how we could move this forward. Particularly, they said, you really want to validate that there's no impact on the ultimate cell type function. So the results that I presented on the cardiac muscle cell, that's part of the validation process that these people would need to see before we can move it into humans. But I guess the bigger question is, and this always comes up when I talk to the patent office, is what do you want to target first, Hai-Ling? What is the lowest hanging fruit? And as a basic scientist, I don't look at anything else. I don't look at anything as more lower hanging than another. I just see the cell tracking platform as being very universally applicable. Some people have identified cancer immune cell tracking as being more, the lowest hanging fruit, whereas they say, regeneration may be farther away, simply because regeneration research is still, I wouldn't say tentative, but it's not as well developed as cancer immune cell therapy. So Eva, I guess the short answer to that is there are still a lot of hurdles. I'll have to find the money to go through all those toxicity studies, but I'm currently getting funds to do these market assessment research and to take it to the next phase. There may be a company, spin-off company, that will come out of all this. I might just license it to some, I don't know just yet, but I'm getting guidance, and I know it's going to be a long process, but ultimately, if before I retire, I could see this going somewhere closer to clinic, I would be very, very happy.
All right. No, thank you very much. By the way, great talk. Yeah.
Thank you.
Okay, thank you. Bruce, please.
Hi there. That was a fantastic talk. I'll second that comment. Really enjoyed it very much.
Thank you.
A couple quick questions on the ferritin. It seems to me if a cell, you program genetically a cell to generate ferritin, if that cell were to die, don't the macrophages pick up that ferritin and the ferritin still ends up in macrophages and thus visible? The notion that it's a marker of viable cells, is that actually true, or is the ferritin get mobilized and taken away and not localize in the macrophages the way an iron oxide particle might?
So that's a very, very good question, Bruce. It's not something that we've looked into. We still don't know how it's recycled precisely, mechanistically inside a cell. We just have evidence that it is being recycled and it's taken away in these lysosomal structures. And the fact that the signal disappeared completely by day 7 meant that it must have been through that route. There might have been other routes, but not ones that I'm aware of, right? That the natural recycling. Whether or not there could have been dead cells that release our manganese ferritin particle into extracellular space and then macrophages come along to take them up, that would depend on when the cells die. And I mean, ferritin circulates in our blood serum, but not in a very high amount, right? The ferritin that circulates in blood serum, when we look at iron, the iron content is relatively low compared to the iron content within ferritin inside of cells. So are there changes in the metal content when these ferritin proteins are released out of a cell, whether it's through natural mechanisms or through cell death? I'm not sure, but it's something I certainly need to look more into.
Sure. The second question is on the so-called Bright-ferritin. I will admit I might have drifted out and missed the critical point. It seems like it's a game you're playing with the use of an exogenous contrast agent.
Yes.
Is there something novel about the form of ferritin that you're programming? And what can you tell us about the contrast agent that you're injecting? So it's not just the expression of the ferritin that's the key, it's the ferritin, and you've designed a contrast agent that is taken up intracellularly and binds to the ferritin. Like I just wasn't quite sure what the story was.
So the ferritin, so the manganese contrast agent was very simple. We have not gone on to make a specific manganese contrast agent just for this. It's simply manganese chloride. And the ferritin, the way that we program it into the DNA is we make sure that it's targeted at a safe region. So we target it within the AAVS1 locus to make sure that it's not interfering with any other gene expression within the cell. Furthermore, that's one mechanism of ensuring no off-target effect by putting it into a safe harbor locus. The other thing that we do is we don't overexpress ferritin by a huge amount. So when you look at the literature where they do report, iron ferritin cell tracking, a lot of those papers overexpress ferritin by quite a large amount. I'm just recalling like at least a factor of 20, whereas in our case, we're only overexpressing ferritin by a factor of 2. So given this very conservative approach that we use for the ferritin overexpression, it is somewhat surprising that we are getting such a huge contrast.
So the manganese chloride is binding to this endogenously generated
Yes.
ferritin.
Yes.
Interesting.
That's what we believe to be happening.
Okay. Cool. Thanks.
Great. I think that's a great point. To thank you so much again, Dr. Cheng, for this great talk. Thanks everyone for joining and Eva for the introduction and organizing. And see you again next week.